So first of all, I just really want to thank the organizers for letting us speak. What you're going to see with Nexamine is something probably a little different, both from a business standpoint as well as scientifically than anything else you'll see in this session. From a business standpoint, we're a brand new company that has just licensed the technology out of Johns Hopkins, so we're relatively young, and what I'm going to describe for you, although we're a young company, is not a young technology. It's a technology that's actually been developed over the last 10 years at Johns Hopkins, sort of a little bit in academic stealth mode in the laboratory of Jonathan Schneck, in which he has created an entire system for creating artificial antigen presenting cells, and I'll take you through both what the platform is and from a business standpoint how we expect to use it. How do I forward here? Sorry about that.
Jeff, did you steal the clicker? Oh, here it is. Got it.
Oh, here it is right here. Okay, great. Thanks. So the company itself, the technology we're calling the AIM technology, it has potential in a number of different disease areas, cancer, transplant, et cetera. Could also be used in some ways for protection against biothreat agents. The company itself is going to focus in cancer. Our first product will be a product for the treatment of melanoma, which we call AIM 101. We believe there will be a lot of partnering opportunity for it. In the interest of time, I haven't included a slide on the management team or the clinical and scientific advisory board. We're in a very fortunate position out of the gate to have an extremely experienced management team, including Bruce McCready, who's in the back of the room, who's heading up our drug development, who's been involved in the development of a number of different immunotherapies, including the largest selling immunotherapy for HIV in the world, and a number of both John Schneck's colleagues at Johns Hopkins as well as other places on the academic side are involved in the company as scientific and clinical advisors, as well as Jim Young, who many of you probably know led and was really the brain trust behind MedImmune for the years that they were a growth company right up to when they sold to AstraZeneca. So we're well positioned from a team standpoint. In terms of the technology, just let me remind you what I think most people in this room already understand very well, that is that natural antigen presenting cells play the central orchestrating role in the cellular immune response, whereby anything that the body needs to get rid of and to monitor from a cellular immune response, whether it be external antigens, bacteria, and whole bacteria or viruses, all the way through to tumor cells and necrotic cells, antigen-presenting cells, take up these materials, process them, present them as antigens in the classic immunology signal 1 on, for example, a dendritic cell, and then along with that on the antigen-presenting cell is signal 2, which provides the instruction for what the cell should do. Now, in the healthy individual, as these antigens are taken up and processed, they are presented alongside what is typically a positive co-stimulatory molecule that then interacts with a naive, for example, cytotoxic T cell, but this could be essentially any T cell in different scenarios for different T cell types. And in the case of cancer and in the case of cytotoxic T cells, what you get from the combination of this presentation is an activation and proliferation of that cell until they become active and in many cases at least small percentage points, and in some cases very large percentage points of the T cells that are in the body as they attack the individual disease entities. In this case, what we're showing here is either an infected cell or a tumor. Now, in a healthy individual, this works well. It's actually a balanced response, so that what you get is not only upregulation, but as the disease is addressed, or the foreign antigen is addressed, then you get negative regulation of these T cells as well, so that they go back down and you have a healthy homeostasis. However, it is often the case in both infectious disease and particularly in tumor growth that the tumors themselves exert influence on this process by secreting cytokines that ultimately downregulate this process, go back and downregulate the antigen-presenting cells so that they present not only upregulating signals but downregulating signals like PD-1, which ultimately shut down this tumor CTL response, and then it leads to what's called tolerance or exhaustion, and you end up with inactive CTLs, and ultimately the tumor's allowed to grow. This is a huge problem in developing cellular therapies, because in cellular therapies you have two issues. First of all, if it's a natural cellular therapy you're dealing with, which everything to my knowledge that's in development currently, you're dealing with natural cells that have started out either in a patient or in someone else. If it's a patient, you have the issue of, do you have healthy cells to start with? If you're doing ex vivo therapy, you also have the issue of, once you do whatever you're going to do with the cells and then put the cells back in the body, how healthy are the cells? How do you deliver them to the site? How do you keep them healthy and engaged while they get to the site of action? And then ultimately, you have the issue of, once they're back in the body, you still have to deal with this downregulation issue. So the beauty and really the simplicity of the concept of an artificial antigen-presenting cell is that you can actually, in a pharmaceutically completely controlled and designed way, create a cellular therapy that you know all the definition of all the components of. You know the definition of the matrix that you've used to create the cell. You know the definition of the molecules that you've used to do the antigen stimulation, the antigen presentation and the stimulation. And then ultimately, because this is an artificially pharmaceutically constructed entity, it's basically a very large and relatively complex NCE, you also design it in a way so that it will not respond to the downregulation that you see in tumors or other diseases. So the technology itself, as I mentioned, is very well developed. It's been developed at Johns Hopkins School of Medicine for about 10 years. The university has done an outstanding job, actually, of the intellectual property protection. There's a portfolio of about 24 patents, 7 of which are issued in the U.S. There's also issued patents in a number of other countries around the world. All of these patents deal with a concept of taking 3 fundamental components that you would see in a natural antigen-presenting cell. The cell itself, in this case, is synthetic matrix, and the patent portfolio is written in such a way that you can essentially use any synthetic matrix, the antigen-presenting molecule and the co-stimulatory molecule, and then present those in a fashion that allows you to use them therapeutically. Just very quickly, there's a number of different ways you can use this platform to stimulate either in cancer a response against a tumor or in using downregulatory molecules in cases of autoimmune disease, for example, where you're trying to knock out a particular T-cell population. I'll kind of skip the plan, quickly just say that we're in the development of our first product for the treatment of anti-melanoma. There's a number of very good, both published and unpublished studies, including this one showing, for example, that in this tumor model you can actually completely obliterate tumors that have been established in some cases in the numbers of hundreds and hundreds of tumors inside the lungs of these animals that have been injected with tumors in the appropriate T-cell, and with a single antigen response to one of these artificial antigen-presenting cells, you completely eliminate the tumors. So with that, I will be happy to answer any questions that folks have afterwards, and would be delighted to talk to folks that are interested in the technology. Thanks a lot.
Just one question.
Sure. Yeah.
You're setting up basically a platform company here as well,
Yeah.
while developing. So are you planning on also out licensing and creating for others?
Yeah, so on this slide here, which is sort of the opportunities, we're focused here in the cancer space, but we see already there's data, for example, around treatment of CMV and post-transplant and such things, so we'll partner around those. Great. Thanks a lot.